Hello, ISMB, and thank you for coming to my virtual poster. My name is John Bachman. I'm a fellow at the Lab of Systems Pharmacology at Harvard Medical School, and I'm presenting work by myself and my colleague Ben Gyori to develop a self-updating and self-testing causal model of COVID-19 drug and disease mechanisms. So we're aiming to address a challenge that applies across the biomedical sciences, which is how scientists can not only discover but also interpret newly published mechanistic findings. This is a particularly acute problem in COVID-19 research, where there is a deluge of new findings being published daily about the virus, but all of that information has to also be interpreted in the context of background knowledge about cell biology and immunology. Our approach to this problem is to use text mining along with a process we call knowledge assembly to build a causal model of COVID-19 molecular mechanisms, and then use this model to systematically identify possible mechanisms of action of drug repurposing candidates. To build the model, we use a system we've previously developed called INDRA, which runs 6 different relation extraction tools on several streams of publications as soon as they appear. INDRA normalizes the output of these different machine reading systems into a common format, corrects systematic reading errors, aligns the relations against each other to identify overlaps, and estimates the overall reliability of each extraction based on the system that extracted it, its frequency in the literature, and its overlaps with other relations. This process, which we call knowledge assembly, is somewhat analogous to how in next-generation sequencing, many short, potentially noisy, and overlapping reads are assembled into a model of the underlying sequence. Since this process is fully automated, we can use it to continuously update disease and pathway models based on newly published findings. This is implemented in a software platform that we call EMMA, which stands for the Ecosystem of Machine-Maintained Models with Automated Analysis. So EMMA not only uses text mining tools to extract new knowledge, that's the machine-maintained part, but it also adds important context to these findings in 2 ways. First, through the knowledge assembly process, the system can determine whether a newly extracted mechanism is already known or overlaps with a mechanism that is already known. And second, the system can determine whether that new mechanism changes the behavior of the causal model implied by that knowledge. So this is the automated analysis part. One way that could happen, for example, is that a new finding could serve as a missing link between other facts that are already in the model that together form a novel causal path that can explain an observation that was previously unexplained. So in this way, models in EMMA are both self-updating and also self-testing. It's also worth noting that in addition, scientists can perform custom queries against one or more models in EMMA and be notified when these queries yield new results. So the causal analysis process that I mentioned is performed by translating the relations in the model into networks with different causal semantics, such that test conditions can sometimes be satisfied by certain network representations, but not others. And these network representations include unsigned directed graphs, signed directed graphs, pi-bell networks, and also the influence map of a rule-based executable model. So EMMA makes the results from all of these network types available so that they can be compared and analyzed by the user. The EMMA website currently contains 12 different models, each of which focuses on a particular disease or pathway. The scope of each model is defined by a combination of literature search terms and lists of genes that are known to be relevant to that pathway or disease. So one of these is a model that is focused specifically on COVID-19, and it is based on information extracted from the COVID-19 literature corpus, virus host protein-protein interactions from databases and recent publications, and a handful of other sources. The COVID-19 model has been running since late March and currently has about 169,000 unique interactions. So we use this model to find causal paths that link specific drugs to their observed or hypothesized inhibitory effect on SARS-CoV-2 or other relevant coronaviruses. So we use 2 different sources for these drug virus observations. The first are a set of drugs that we manually curated from 4 in vitro drug repurposing screens against SARS-CoV-2. And the second is a set of drugs that have been identified as currently under clinical investigation for COVID-19 efficacy. These are obtained from MITRE's COVID-19 Therapeutic Information Browser, which is linked in the poster. So for example, the MITRE corpus lists camostat as potentially relevant to SARS-CoV-2 and other coronaviruses. And Emma finds a causal path linking the drug to the virus via the drug's target, TMP-RSS2, which is the co-receptor for SARS-CoV-2. This is obviously a simple example, but these paths can actually be more complex, and for the empirical hits from in vitro drug screens can highlight interesting areas of host virus biology to explore. Of course, text mining systems make mistakes, so the user can drill down into the sentence level information for any particular causal link in a path to review it, and if necessary, flag an extraction as incorrect. And these curations are propagated back into the system, and paths are updated with this new information as part of the daily cycle. So in conclusion, we built a system called Emma that uses text mining to extract new findings every day, and then uses those new findings to search for causal explanations of empirical observations. And we've used this to build a mechanistic model of COVID-19 mechanisms that can systematically generate hypotheses for the mechanisms of action of COVID-19 drug repurposing candidates. That's all, and thanks for listening.